So I'm happy to be here to talk about some of our work at a public health agency in the US on detecting and differentiating abrin and ricin. So I'm sad to tell you, abrin and ricin are plant toxins. So they're plants, right? They don't bite, they don't sting, they just kind of hang out and do plant things. But I think they're pretty cool toxins nonetheless. So wanted to start with a bit of an explanation on ricin and what it is. Do I need to point this in a particular way? Ah, yes, that works. Okay, excellent. So ricin is a protein toxin that comes from a plant. So it comes from the castor bean plant, which is called Ricinus communis. And the plant produces these really pretty seeds that are just absolutely gorgeous. And so these are castor beans, they're seeds. They're produced in these kind of hairy type of pods. And you can see there's some pods that are split open here. So the pod is kind of hairy and it's protecting these really pretty beans. So ricin is a protein toxin that's produced and lives kind of within these beans. It's a 64 kilodalton protein, so pretty big compared to a lot of the things that we've heard today. And it's made up of two chains. So there's an A chain that's about half of it, at 32 kilodaltons. And the A chain has enzymatic activity, which we'll get to in a minute. The B chain is about the same size, also about 32 kilodalton, but the B chain doesn't have any enzymatic activity. Its job is to bind receptors on the target, on our cells inside of our body to deliver the A chain to targets. The B chain's heavily glycosylated, and this glycosylation is important in it being toxic to humans. These two chains, the A and the B chain, are disulfide linked. And both chains are needed to be linked together in order for it to be toxic. So just one by itself is not toxic. So it turns out there's a similar protein called abrin. You'll notice abrin's got a lot in common with ricin, also a plant toxin. Its source is from Abris precatorius, and it's called the rosary pea, and it's this red bean-looking thing right here with a black spot on it. It's used in a lot of decorative jewelry. It's also actually really pretty. You see it looks very different from what I just showed you, the rice and the castor bean, right? But they're still both beautiful beans. And so they have these plant toxins in them. Abrin's incredibly similar to ricin in what it does inside the human body. 64 kilodaltons, we've heard this before. Two chains, heard this before. The A chain has enzymatic activity. B chain binds receptors. B chain's glycosylated. Chains are disulfide linked. So similar to ricin. Both chains needed for toxicity. All of this, right? Incredibly similar. Turns out both have the exact same enzymatic activity. And what they do is they depurinate adenosine on 28S ribosomal RNA within our bodies. So what is depurination? So what I'm showing here is kind of a blow up of the portion of 28S ribosomal RNA that ricin attacks. So we've got bases here. We've got a GA structure here that's really important. What happens is ricin or abrin will come in and it will clip the adenine off of that GA portion. So it's a very minor change that's occurring on 28S ribosomal RNA. It's just clipping off adenine. Adenine's 118 daltons. Really very minor change. But it turns out it has a very large detrimental effect for us. So 28S ribosomal RNA, we have it in our bodies. We love it. It works great. In order to make proteins, there is elongation factor 2 that binds on that GA portion that I just showed of 28S ribosomal RNA. EF2 binds. This is great. This is what produces protein synthesis, and this is what keeps us alive. Okay. But now if we take ricin or abrin, the ricin or abrin will go in and it will depurinate our 28S ribosomal RNA. It'll clip off that adenine. Now EF2 cannot bind to 28S ribosomal RNA. Very minor change, very big consequences. Since EF2 can't bind, this is what halts protein synthesis. And it's that halt of protein synthesis that results in cell death. So these are things that we know about ricin and abrin and the effects that they have on the human body. So I come from a public health agency. We're very interested in trying to design assays to detect if people have been exposed to ricin or abrin. And so that's what we've done. So we took the knowledge that we had about ricin and abrin. We wanted to design an activity assay to detect if these toxins are active and if they're toxic, right? So we know abrin and ricin attack 28S ribosomal RNA. What we've done is we've designed a synthetic RNA target, which is here. It's 14 bases, GC-GC base paired, and the GA-GA tetraloop is present there. When ricin or abrin to come into contact with that, they will depurinate this adenosine, just like it does inside the human body. So why do we want to work with this synthetic RNA target rather than working with 28S ribosomal RNA? Because I want to do this detection by mass spec. And detecting this by mass spec is a lot easier than detecting 28S ribosomal RNA. Okay, so this is how it works. If we have ricin or we have abrin, we add in our RNA substrate, our mimic that I just showed. And if the toxin's active, it will depurinate and it will cause that adenine to just kind of float off. And we have our depurinated substrate. What we then do is take the substrate and put it into our mass spectrometer. So not the toxin, but the substrate itself. And we get two peaks if ricin or abrin is present. The black, larger in molecular weight, is the intact substrate. This just tells us this is your RNA substrate you can detect. If this peak is present, though, at 118 Daltons less, that's corresponding to the depurination of our intact substrate. So that's telling us ricin, abrin, or something is present that's depurinating our substrate. So that's what we're looking for. But when we start to try and work this in complex samples, everything works great in buffer, but this just really did not work well. So we recognized immediately we needed some kind of affinity purification in order that was going on before we got to our activity test. So we tried a lot of things. What we ended up settling on recently was a magnetic bead that's coated with a glycoprotein. So we take that magnetic bead, we add it into our sample, which may or may not contain abrin or ricin. If abrin or ricin is present, it binds to that glycosylated protein. We can wash the beads, wash everything else away. That's when we can add in our synthetic RNA substrate. We let it react with the toxin that's on the beads, and then we do that measurement by mass spectrometry. So it's sort of a two-part assay in order to detect and differentiate. So the first part of this, which I've just shown you, this is detection of ricin or abrin's enzymatic activity rather than the toxin itself. And this allows for an assessment of toxicity. It allows us to determine whether or not that abrin and that ricin has the potential to harm a human being. And that's really important for public health purposes. But I've just talked to you about how abrin and ricin are similar, right? They have the same enzymatic activity. So in order to differentiate them, we have a second part of this. And that second part gives us our accuracy. So there are other ribosomal inactivating proteins that have the same enzymatic activity as abrin and ricin. So in order to differentiate ricin from abrin and from those other proteins, we have a second technique where we go in and we take a look at the amino acid sequence of the toxin. And that tells us if it's abrin, if it's ricin, or if it's something else. So the assay works like this. We have our sample. We do our affinity extraction with those magnetic beads. We add in our RNA substrate. We let that react for a little bit, and then we take that substrate out to the mass spec. That gives us our activity or our toxicity assessment. If we get a hit, if it's positive, it's active, we have something there, we want to go back, take a closer look at it. We go back then not to the sample, but to those magnetic beads, the toxins on those magnetic beads. We digest them with trypsin, and we do mass spec of the tryptic fragments. And what I'm going to show today is the peptide mass fingerprinting of that although we've also been known to do a lot of LC-MS/MS, but today I'm just showing the peptide mass fingerprinting. And that gives us some sort of information on the structure of it, a little bit of information on the amino acid sequence to help us determine ricin, abrin, or something else. So the peptide mass fingerprinting part, just wanted to show a little bit on how that works for people that may not know. So in this case, I have my magnetic bead and ricin is attached to it. We will hit it with trypsin. This results in a series of peptides ending in lysine or arginine. And those peptides are unique, actually, for ricin. We then do MALDI mass spec on that, and we get sort of what's called a fingerprint, right? So it's a readout. All of our peptides that are here, we're getting peaks for in our mass spectrum, and that's sort of a fingerprint for that particular digest. It looks different when we do a digest of abrin. So here's abrin. We digest it with trypsin. We also get a series of peptides ending in lysine or arginines, but the amino acid sequence is different. So the peptides themselves are different. Run that on the mass spec, and we get a fingerprint, if you will, of what abrin looks like. So what we can do is we have digested abrin and ricin a number of times. We have a good feel for what those two fingerprints look like, right? So the top is ricin, the bottom is abrin. You can see in this example how they look different. If we then have a sample, we can take our unknown sample and we can try and match it. So we can try and match it here. We see that it's not a good match. That tells us it's not abrin. Okay, that's fine. So we take our unknown sample. We now try and match it against the data that we have from ricin. It matches. This tells us our unknown sample is now ricin. So that's essentially how it's done. So I just wanted to show a little bit of some actual data that we've been generating. This is abrin and ricin that we've spiked into talcum powder to be able to detect it. The spectrum all the way on that side is the blank. And so what we're looking at is a peak of our intact RNA substrate. It hasn't been modified by a toxin because there's no toxin there to modify it. The spectrum in the middle is, oops, sorry, I went back. I was looking for the pointer. Pointer doesn't work. Abrin. So here's our intact substrate that's here. And notice we now have a new peak that's grown in. That peak is the depurinated substrate that I talked about. So basically what this peak tells me is that I have either abrin or ricin. Notice how abrin and ricin give the same, I mean, they look identical, right, in the enzymatic activity assay. So we would see this, we would say, okay, it's either abrin or ricin, but we're not sure which. We go in, we do our digest, and we take a look at our digests on the mass spec. So many of you may not be completely familiar with mass spec, but hopefully you can see and see well enough to notice that the two have definitely some similarities, but they're not identical. So you can see that there's something here that's going on that wasn't there before, some other changes that are present throughout. So a lot of it's similar because when we do our tryptic digestion, we're digesting the beads plus the toxin. And the beads are the same in both cases, but the toxin's different. So we just go in and we look at our maps and we can be able to tell if something's abrin or ricin. So that's essentially what we've been doing. We've been trying to get the assay validated so that we can roll it out to our state public health laboratories in the US who are very interested in being able to detect abrin and ricin for public health purposes. So in summary, mass spectrometry is a useful tool to detect and differentiate ricin and abrin, and a single assay detects both plant toxins via their enzymatic activity and differentiates them based on their amino acid sequence. So that's what I have. Hopefully I did okay on time.